We'll start with another chapter for today, that is your cell cycle and cell division. Okay, Makha? Now moving ahead. Now, children, the sequence. Now, what do you mean by a cell cycle? It's nothing but the sequence of event which a cell duplicated its genome. Am I clear to you? Not only that, the synthesis of the other constituents, okay, of a cells and eventually divides into two daughter cells is nothing but your cell cycle. I repeat again, what is a cell cycle? Okay, it's a sequence of events by which a cell duplicates. Well, one is there, it will give rise to two. Okay, there even your particular other constituents, not only your particular cell, whatever is present inside the cell is also duplicating. Am I clear to you? Also, along with the genome, why your hereditary has to pass on from one generation to another generation. Henceforth, the genome will also duplicate, it will also replicate. Am I clear to you? And this is nothing but what, okay? Cell cycle. Okay. Now, the DNA synthesis occurs in one specific stage of a particular division, but distribution of the chromosome in cell complex occurs series of events during the cell division. And that synthesis of DNA you have in one particular point, whereas the rearrangement of the chromosomes, like in your metaphase, it is coming to your center equatorial plate, then it is moving to your particular anaphase in the poles, then finally it is reaching back to your particular telophase where the two cells are replicated. So rearrangement is looking very complicated in each and every cell. Okay, so whereas in interphase you see it's duplicating. Am I clear to you? So that's the point. Moving on to the next one. Human cell divides once in approximately 24 hours. Keep this in your mind, okay? So human cell usually approximately divides within the 24 hours, which may vary in different organisms. In yeast, it takes 90 minutes. Okay, na? Yeast ಅಲ್ಲಿ 90 ಆದರೆ ಮನುಷ್ಯನಲ್ಲಿ ಎಷ್ಟು ಮಕ್ಕಳೇ 24 hours. Am I clear to you? Now, phases of cell cycle. So we'll begin quickly with the phases of cell cycle. Okay, so here you have a particular G1 phase, S phase, G2 phase and M phase. Am I clear to you? That's your mitotic phase. Am I clear to you, children? So usually what is each one is called, we will see in a individual manner. Okay, now in the particular interphase, am I clear to? Basically, a cell cycle consists of two phases, interphase and M phase. So, from here to here, this complete entire structure is nothing but your interphase. Only from here to here is nothing but your metaphase. Am I clear to you? Full. Illinda ಹಿಡಿದು ಎಲ್ಲಿವರೆಗೂ ಮಕ್ಕಳೇ till here. Okay, so basically from here to here, what is it? It's nothing but your interphase. Only this is your metaphase. Okay. Now, in interphase, what happens? It is a phase between two successive M phases. Okay, so interphase lasts up to 95% of a cell cycle. So, which is the longest phase compared to mitotic and interphase? It is always your interphase. Yeah, 95% of a cell cycle is made up of interphase. This phase is usually called as your resting phase. What does it usually call it as? Resting phase. Why is it called as resting phase? This is the time where a cell is preparing itself for what? For the further cell division. Okay, so but during this period the cell prepares itself for the nuclear division by a cell growth. That's what I'm telling you. So the cell prepares for the furthermore division. Okay, so that is what we call it as your interphase. Now moving ahead, the next one is your M phase. Now coming to our M phase, when an actual cell division or mitosis occurs, what happens, children? So there is an actual cell division or a mitosis occurs during what phase? During your M phase. It starts with karyokinesis. First to karyokinesis. ಅದಾದ್ಮೇಲೆ ನಮಗೆ cytokinesis. So keep this in your mind. First always comes your karyokinesis and then comes your cytokinesis. Sweethearts, when I say karyokinesis, it's always your nuclear, and cytokinesis, obviously it is your cytoplasm. Am I clear to you? So it is in your metaphase what happens? Exactly your duplication of chromosome comes. Am I clear to you? It is in your metaphase or a mitotic phase. That's your duplication of chromosome appears. Okay, now the interphase. is divided into three further phases. So we'll first begin with your interphase. Makla innonsali ಹೇಳ್ತಾ ಇದೀನಿ. In interphase, the cell is preparing itself for the cell division or the further particular processes. Now that's called as a makla interphase. And interphase carries about your 95% of the cell cycle. Whereas in your M phase, that is your mitotic phase, here you have your duplication of the chromosome. First ಏನಾಗುತ್ತೆ ಕಂಡ್ರು, ಫಸ್ಟ್ ಯಾವಾಗಲೂ ಕ್ಯಾರಿಯೋಕೈನೆಸಿಸ್, ಅದಾದ್ಮೇಲೆ ಏನಾಗುತ್ತೆ ಮಕ್ಕಳೇ, ಅದಾದ್ಮೇಲೆ ನಿಮಗೆ ಸೈಟೋಕೈನೆಸಿಸ್. ಓಕೆ, ಸೊ ಕ್ಯಾರಿಯೋಕೈನೆಸಿಸ್ and cytokinesis. Now we'll begin with the interphase. Now that means whenever I started with your particular interphase, I'll come back to this. Okay, cell cycle consists of three phases, that is your G1, S, G2 and M phase. Now G1 phase is usually called as your growth and synthesis phase. S is obviously denotes your DNA synthesis phase. G2 preparation for division. M phase includes your overlapping process of mitosis and cytokinesis. That is nothing but your ella nimge prophase metaphase ella baruttala avella ನಿಮ್ಮ M phase alli ide. Am I clear to you? See, this is the one. So in G1 phase, what is happening? Growth and the normal metabolic rules. So this is the place where your first growth phase anta karite. ಏನಂತ ಕರಿತೀವಿ G1 ನ? The G1 stands for your first growth phase. Okay, next comes your particular S phase, nothing but your synthesis phase. What do we call it? As synthesis phase, where your DNA replication happens. Am I clear to you? Whereas what is G2 phase is called as second growth phase, where growth and preparation for the particular mitosis is happening. So entirely, this entire structure, see that blue color itself is denoted by your interphase. Can you see her children? It's completely called as your interphase. Am I clear to you? So we are studying about this particular thing now. Let's start with the G1 phase. Okay, now gap phase 1 begins at the completion of your mitosis. Mitosis with takshana, the first thing which starts is nothing but your G1 phase. Okay, and cytokinesis. Okay, and last up till the beginning of the S phase. So G1 phase, it will begin right after your mitosis. Okay, after mitosis, where in mitosis, once karyokinesis begins, once cytokinesis finishes. Okay, so karyokinesis agi, cytokinesis agi takshana, which is the next one, it is your G1 phase. And last up till what particular phase? It lasts up till your particular what to say, the cytokinesis. Immediately afterwards, what will start up children? It will start up with your G1 phase. So G1 phase will last up till what time? Once your S phase begins, till then you will have your G phase. Andre cytokinesis mugdik takshana, you have your G phase starting up. So G phase will end up when your S phase, that is your synthesis phase, begins. Now this phase is generally the longest of the 4 cell cycle. Remember this. Okay, so one of the longest phase is your particular G1. Okay, now, and it is quite variable in length. During the phases, the cell chooses either to replicate its deoxyribonucleic acid, nothing but your DNA, or to exit the cell cycle and enter into a quiescent state. So then in this particular thing, what will happen? The replication of DNA begins, or else there will be no particular division or nothing. It will be in an empty phase. Okay, now that stage we usually call it as your quiescent state, nothing but your G0 phase. So remember that, nothing but your G0 phase. Am I clear to you? So that is about your G1 phase. So in adult animals, some cells do not divide or may divide occasionally. Now these cells do not divide further and exit the G1 phase to enter an inactive stage called as your quiescent stage, nothing but your G0 cell cycle. Okay, so where there is no cell division further agala, cell division agilla ಅಂದ್ರೆ ಹಂಗೆ ನಿಂತು ಬಿಡುತ್ತೆ the cells. It's not compulsory that all the cells has to divide. Some of the cell will not divide, and those particular cells will enter into a stage called as your quiescent stage, nothing but your G0. Moving on to the next one is your S phase. What is it, Makla? S phase. Here, the replication of the chromosome is restricted to one specific portion of the interphase called as your S phase, where exactly your DNA synthesis phase happens. In G1, there was a growth. So in S phase, there is a DNA synthesis, which typically lasts about how many hours? 6 hours. It will last about just 6 hours. In mammalian cells, the start of a S phase, the actual initiation of the DNA synthesis takes place several hours after the cell has committed to carrying out the DNA synthesis. So in mammalian cell, the S phase, whatever is beginning up, is lasting for many hours because there should be a synthesis, right, of the DNA. Henceforth, am I clear to you? Now, during S phase, each chromosome replicate exactly once to form a pair of physically linked sister chromatids. ನೋಡಿರ್ಬೋದು ನೀವು chromosomes form ಆಗೋದಕ್ಕೆ. Am I clear to you? So they are called as your sister chromatids. Yes or no, children? So that is what we are speaking about. In animal cells, the pair of centrioles is also duplicated during S phase. So if this is your cell, so you will have your particular centrioles. Yes or no, children? So that will duplicate. So one will move towards an opposite side. Yes or no? So even the centrioles will also duplicate during this S phase. So basically this is the phase where entire, what to say, the duplication or the replication of the DNA is happening. Next one is your G2 phase. What is it, Makla? G2 phase. Now, the portion of the interphase that follows S phase is called as your gap phase 2. Am I clear to you on that? G1 ಆದ್ಮೇಲೆ S ಆದ್ಮೇಲೆ G2. So S phase comes in between G1 and G2. Am I clear to you? So S phase comes in between G1 and G2. Am I clear to you? So some of the cells can exit the cell cycle from G2 phase just as they can from G1 phase. ಅಂದ್ರೆ G1 phase ಆಗಿದ ತಕ್ಷಣ ಎಕ್ಸಿಟ್ ಆಗ್ಬಿಟ್ರು, ಸೆಲ್ ಡಿವಿಷನ್ ಮಾಡಲಿಲ್ಲ ಫರ್ದರ್, ಅದನ್ನ ನಾವು ಕ್ವಿಸೆಂಟ್ ಸ್ಟೇಜ್ ಅಂತ ಕರೆದ್ವಿ. ನಾಲ್ಕೈದು ಮಕ್ಕಳ. ಸೊ ಅದೇ ರೀತಿ G2 ಆದ್ಮೇಲೆ ಈವನ್ ಆಫ್ಟರ್ G2 some of the cell instead of going to your mitotic phase they will exit the cell cycle. Okay, now combination of these three phases are usually known as your interphase. ಯಾವ್ಯಾವುದು? G1, S and G2 phase. Okay, so G1 phase, G2 phase and S phase. In G1 phase, if at all they are not dividing, what they enter into? G0, the quiescent stage. Am I clear to you, Makla? So that is about this. Moving on to the next one is your M phase. Okay, so M phase includes the overlapping process of mitosis and cytokinesis. So here what happens? You have mitosis, that is nothing but your where you have your karyokinesis beginning up. Yes, initially you saw all the duplication and everything coming into the middle, rearranging themselves, the chromosomes, chromatids. Okay, and finally you have your cytokinesis phase. Okay, so this is how exactly they look like. Okay, so this is your prophase, metaphase, where you have they aligned in the center, where they are moving towards the opposite poles. Okay, which one? Anaphase, where you have only sister chromatids moving up, and here you have your cytokinesis happening. Okay, so that is about the cell division. Now, cell cycle is the event that makes a new cell by cell division through a 6 processes, which are those. First one is interphase, which you have already dealt with. Next one is mitosis, prophase, metaphase, anaphase, telophase and cytokinesis. So all these are the processes where you require for a cell to undergo your cell division. Okay, now first one will start up with what, Makla? Mitosis. Okay, now nuclear envelope breaks down into vesicles during prophase and reassembles when the vesicle fuses to form an envelope back to the telophase. ಏನಂತ ಹೇಳ್ತಾರೆ ಮಕ್ಕಳೇ? At the starting of the interphase, what happens? The nuclear envelope so present, that is especially in the prophase, what happens is the nuclear envelope will break down. Am I clear to you? It will break down. And what this will form? This will form your vesicles. Once they form a vesicles, okay, they will be like that. Up till what phase? Up till your telophase. Break ಆಗಿ ಏನಾಗುತ್ತೆ, ಒಂಥರ ವೆಸಿಕಲ್ಸ್ ತರ ನಿಮಗೆ ಫಾರ್ಮ್ ಆಗ್ತಾ ಇರುತ್ತೆ, ಒಂಥರ ರೌಂಡೆಡ್ ಲೈಕ್ ಅ ಬಾಡೀಸ್ ಲೈಕ್ ದಟ್. Am I clear to you? Now when will they rearrange back? You remember in the telophase, again your nuclear envelope will reappear back in each cell. So how did they reappear back? Because these vesicles joined again. Am I clear to you? So these vesicles will join again and make the nuclear envelope reappear back. Now, cytokinesis, division of the cytoplasm between daughter cells, lasts up to the last stage of the cell division. What is the significance of the mitosis? Children, usually it is required for your growth. Yes or no? So if a cell has to, or organism has to grow, your cell has to divide, your cell has to multiply. Only then it forms a tissues, organ, and organ system. Okay. Next, immune response, like your B and T lymphocyte, during which immune response is dependent on mitosis. Yes or no? We require your B lymphocytes, T lymphocytes in our body. So it has to be produced frequently. So the cell division has to be there for the immune response. Replacement and repair. Something particularly in your body, something tear of a tissue is there. Anywhere in your body, okay, you have fallen down or something like that. Yes or no, children? So during that time, the replacement of the cell is important. Am I clear too? But replacement, remember, as the person is growing older and older, what happens? Your thymus gland, which is actually producing B and T lymphocytes, is degenerating. Yes or no? Which one? Your thymus. So that time what happens? The replication for the B and T lymphocytes will not be, so immune system will be less. Okay, and not only that, the older the person becomes, the replacement capacity or the mitosis capacity will also start reducing. Am I clear to you, children? Now, asexual reproduction. I'm sure you have already studied this in your particular, what to say, reproduction in organism. Okay, so where you had budding, yes, you had your offset, stolen, runner. Okay, so all these things were growing another particular plant. How? Again, there was a cell division happening. There was a new cells which were forming, fragmentation. It was forming another cell. Yes or no, children? So when they are keeping on forming and forming the new cells, you require mitosis to begin. You require mitosis to happen. So this is called as your the significance of mitosis. Am I clear to you, Makla? Now moving ahead. The first we will start up with, this is how is your interface. So that is what we were learning. There you have your chromatin network. See, children, you have a chromatin as just like that. Am I clear to you? They are very thin, delicate. Okay, so you do have your particular what to say, the nuclear envelope is still in contact, nucleolus is there, okay, plasma membrane is there, so centrioles are also there. So these are the particular this is how your interface looks. Okay, now we'll start up with the prophase. Prophase is the first phase of mitosis, followed by your G2 phase. It involves the following events. What are those? Initiation of condensation of chromosome materials. Meaning to say, your chromatin network has started to condense now. Am I clear to? They have started to form your little thicker substances. Okay, movement of the centrioles towards opposite poles. You just saw the centrioles form, replicated centrioles. Now, what happens in the prophase? The replicated chromosomes will, centrioles will move towards your opposite poles. Okay, at the end of the prophase, endoplasmic reticulum, nuclear membrane, Golgi complex disappears. So all these particular organelles, whichever is there, will start disappearing in the prophase. Okay, Makla? Now, see, the centrioles is moving towards opposite pole, Almendra. Not really. They are moving towards an opposite pole. I'll just show you so that it will be easy for you. See, here ಏನಾಗಿದೆ ಮಕ್ಕಳೇ, ಇಲ್ಲಿ replicate ಆಗ್ಬಿಟ್ಟು ಎರಡಿದೆ ಅಲ್ವೇನ್ರೋ. Now see, here it is a chromatin, okay, network. Okay, don't forget this. Now see here what has happened. Your centrioles are started moving to opposite poles. Your chromatin network has started arranged in a chromosome manner. Am I clear to you, okay? So this is about your particular prophase. Now moving ahead, I want you to look at the diagram. Okay. See, this is how exactly a prophase look in a microscopic view. Okay, ಇದೇ ರೀತಿ ಕಾಣಿಸೋದು ಕಂಡ್ರೋ ಇಲ್ಲಿ ನೋಡ್ರಲ್ಲಿ ಒಂಥರ ನೆಟ್ವರ್ಕ್ ತರ ಇರುತ್ತೆ ಈ ತರ. Okay, so these are the prophase. This is how your prophase looks in your cell microscopic view. Okay, this is how it looks. Now, what's the differences? Always keep this in your mind. This is your animal cell and this is your plant cell. What's the difference? You don't have a cell wall. In what? In animal cell. Am I clear to you? So that's your animal. The first pick is your animal cell. The second pick is your plant cell. Next, moving ahead, the next phase is your metaphase. Now, children, in metaphase, it starts with the completion and the disappearance of your nuclear membrane. So, nuclear membrane ಏನಾಗ್ಬಿಡುತ್ತೆ complete ಆಗಿ disappear ಆಗ್ಬಿಡುತ್ತೆ. Okay na? Now, the most suitable stage for the study of morphology of the particular chromosome, it involves the condensation of the chromosome materials. What it will involve, children? It involves the particular condensation of the chromosomal materials. Okay. into the compact and the distinction of the chromosome made up of your two sister chromatids. Am I clear to you? What is it made up of? Two sister chromatids attached with what? It is attached to your particular spindle fibers with kinetochores. Am I clear to you? So once again I repeat it. Condensation of the chromosomal materials into the compact of the distinct chromosome made up of your two sister chromatids. So two sister chromatids irutte. Then you will have your what to say, the kinetochores right in the center where you have your centromere. Okay. Now chromosomes are arranged at the center of the cell is called as your metaphase, where it is aligned in your equatorial plane. See, this is where exactly it will be placed. Here you have your centromere. Okay. So a chromosome will be aligned right in the center. Okay, children. Now in this phase, what you have to observe is they have your spindle fibers coming up. Okay, so of course it has started already in your particular prophase, but here it is completely distinct where your centromere is attached onto the spindle fiber and they are arranged right in the center. So this is how it usually looks like. Animal cell and particular plant cell. Usually the animal cell ಅಲ್ಲಿ ಹಿಂಗೆ ನೀಟಾಗಿ ಕಾಣುತ್ತೆ. ಆಮೇಲೆ I'm sure it has seen already in onion peel, okay, where you have your metaphase like this. Animal cell and plant cell. Now next one is your anaphase. Now in anaphase it involves the following step where splitting of each chromosome at the centromere and two sister chromatids. Okay, so alvaga nimge enagittu, you had it like this. Now what is happening? They're splitting up like this. Am I clear to you? Now two chromatids starts moving towards your opposite poles. Where do they move to then? The sister chromatids, they move towards your opposite poles. Am I clear to you? See? This is how they usually move towards your opposite chromosomes. Which one, children? In your anaphase. Okay? So that is your animal cell. This is your plant cell. The next particular thing is children, telophase. Okay, now coming to your telophase is last stage of mitosis, which involves chromosome reach at opposite poles and lose its identity as a discrete unit. Andre ಏನಾಗುತ್ತೆ opposite pole cytokinesis wherever somewhere it is starting up, yes or no, in the telophase, because it's the last stage. Now nuclear member reassembles around the chromosome. I told you. What did I say, you children? Usually in the previous one, what happened? The nuclear envelope actually disintegrated and each bit started forming your vesicles. Now darlings, this one here, what happens, will come back, okay, and they rearrange back and again they form your nuclear membrane. Okay, so nucleolus, Golgi complex and ER reappears. So other enagittu initially all these things actually disappeared. ಯಾವ್ಯಾವುದು nucleolus, Golgi complex, ER ಎಲ್ಲ disappear ಆಗಿಬಿಟ್ಟಿತ್ತು. Now telophase ಅಲ್ಲಿ ಏನಾಗ್ತಿದೆ, it is reappearing back. Okay, now finally it will lead to your cytokinesis, where it is your cytoplasm particular division. But remember, before cytoplasm ಏನದು, first to karyokinesis, so then it will lead to your cytoplasm. So sweetheart, first it is your karyokinesis which is taking place, and then it is your cytoplasm division, cytokinesis taking place, where you have your two daughter cells. Now in animal cells, what happens? The appearance of the plasma membrane, furrows in the plasma membrane like this. Okay, furrowing ಆಗ್ತಾ ಇರುತ್ತೆ ಈ ರೀತಿ. Am I clear to you? So furrowing will begin up, so that there will be a particular, what to say, cytokinesis happening that deepens gradually and joins to divide the cytoplasm into two parts. Now in plant cell, wall formation starts at the center, grows outward and meet the later walls. So the formation of the cell wall begins from the formation of the cell plate. So if this is your plasma membrane, here like this furrowing will happen. For what? Animal cell. Whereas for the plant cell, what will happen? There will be like this, the cell plate formation. Finally there will be a cell wall formation. So this is the difference between your animal cell and your plant cell. Aitendra? See, this is the one. So telophase and your cytokinesis. So there is a clear furrowing like this. Okay, see, the nuclear envelope is also reappearing back. Okay, this is how usually your particular telophase looks like. Am I clear to you? So this is how exactly. See, in one day cell and you two nucleus notira. Am I clear to you? So this is how usually a particular telophase looks like. Now, as we have already discussed, but few more things. Significance of mitosis. Mitosis produces a diploid daughter cells with identical genetic complement. Okay, it helps in repair of the cells, especially in the lining of the gut and the blood cells. Very important. Okay, inner lining of the gut has to be repaired. Meristematic division at the shoot apical and the root apical meristem, and also in your cambium, you require your mitosis to happen. Now we'll begin up with your meiosis. Children, the cell division that reduces the number of chromosomes into half, okay, and results in the production of a haploid daughter cell is usually called as meiosis. Mitosis is equational, meiosis is your reductional. But again, you have a difference. What is it? Meiosis 1 and meiosis 2. Okay, it is only your meiosis 1 which is the reductional division. Meiosis 2 is your equational division. Already human reproduction you nodirtira clean age. Oogenesis ali. Am I clear to you? So in oogenesis, your particular egg cell will first undergo meiosis 1. Once your primary oocyte is formed, after that what it is undergoing? Okay, so it is undergoing your meiosis 2. So meiosis 1 is nothing but in makla, it is your reductional. Meiosis 2 is your equational. So keep this in your mind. Okay, it helps the production of haploid phase of life cycle in sexually reproducing organisms. Okay, now it includes following events. Yeah, we have the two sequential cycle of your nuclear and the cell division called as meiosis 1 and meiosis 2, but a single cycle of the DNA replication. Okay, children, so how many phases it contains? Meiosis 1 and meiosis 2. Am I clear to you? Now it involves in a repairing of homologous chromosome and recombination of them. Okay, pairing of homologous and the recombination has happened in where? In meiosis. Pairing and the recombination, the paternal and the maternal chromosomes will overlap with each other and exchange of the genes will happen. 4 haploid cells are formed at the end of the meiosis 2. Remember this, end of the meiosis 2, how many cells will be formed? 4 haploid cells. Okay. So meiosis 1 includes so many phases: prophase 1, metaphase 1, anaphase 1, telophase 1. Meiosis, everything will be in your phase 2. Okay. Now we'll start up with your first one is your prophase 1. Okay. Now in the prophase 1, it contains many particular stages. Now the first one is, children, it's your leptotin. In leptotin. Now children, in the leptotin, the chromosome becomes distinct and visible under microscope. Okay, so prophase ali ನಿಮ್ಮ leptotin is your first stage. Okay, where entirely what to say, it will start your chromosomes will become very much visible. Compaction of the chromosome continues throughout the leptotin. ಅಂದ್ರೆ compaction ಅಂದ್ರೆ ಏನು ಒಂತರ they are condensing somewhere. Okay, next stage is your zygote. What is it, children? Zygote. Am I clear to you? So in the zygote stage, chromosomes start pairing together. So this is where a synapsis will happen. Pairing means what, Makya? Synapsis. The paired chromosomes are called as homologous chromosome, and the particular pairing, okay, point we usually call it as your synaptonemal complex formed by a pair homologous chromosome is called as a bivalent or a tetrad. Okay. Now next stage is your Pachytene stage. So zygotinal ಏನಾಯ್ತು synaptonemal complex. Pachytene ಏನಾಗುತ್ತೆ crossing over between non-sister chromatids of homologous. Okay, so there is a crossing over like this. There is a crossing over. Am I clear to you? So the crossing over of the homologous chromosome occurs for an exchange genetic materials. The crossing over is an enzyme mediated process which involves an enzyme recombinase. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ ನಿಮಗೆ ಪರ್ಟಿಕ್ಯುಲರ್ ಆಗಿ ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಆಗುತ್ತೆ. ಕ್ರಾಸಿಂಗ್ ಓವರ್ ಗೆ ನಿಮಗೆ ಒಂದು ಎಂಜೈಮ್ ಬೇಕಾಗುತ್ತೆ, ಅದು ನಥಿಂಗ್ ಬಟ್ ಯೋರ್ recombinase. Next one is your diplotin. It is usually recognized by dissolution of synaptonemal complex. Synaptonemal complex will dissolve over here and tendency to separation of the bivalent except at the site of a crossing over. Now this forms like an X structure called as chiasmata. ಆ ಪಾಯಿಂಟ್ ನಾವು ಯೂಶಲಿ ಏನಂತ ಕರಿತೀವಿ, chiasmata ಪಾಯಿಂಟ್ ಅಂತ ಕರಿತೀವಿ. ಓಕೆ, ಸೊ ಲಾಸ್ಟ್ ಒನ್ ಇಸ್ ಯೋರ್ diakinesis. It is marked by a terminization of the chiasmata. So if this one point is chiasmata, it will start terminating. Okay, so there will be a terminization of your chiasmata. The nuclear membrane breaks and the nucleolus reappears. So this is where your nucleolus reappears. So keeping again in the point, makle, first one is lactotine, zygotine, pachytine, diplotine, diakinesis. DD last one ಇದೆ. Am I clear to you? So remember like this, diplotine, diakinesis at the last. So first one is what? Lactotine. Then is your zygotine, pachytine. Lactotine, it is only visible of your chromosome material. Okay. Next zygotine, it is your synaptonymal complex. Pachytine, where your particular recombination happens, crossing over happens. Am I clear to you? Diplotine, chiasmata formation. Diakinesis, terminalization of the chiasmata. So these are the important points. Next one, we'll start up with the metaphase 1. Okay, now in metaphase 1, what happens children? In metaphase 1 children, usually the bivalent chromosome align at the equatorial plate. Okay, now obviously all the chromosome where it will come in the equatorial plate. Okay, now moving ahead, microtubules from the opposite poles of the spindle fibers get attached to the pair of homologous chromosomes. So right in the center they arrange. So in the center they usually, in the center of the plate they usually arrange. Okay, Anaphase, what will happen? They will usually split up and move towards your opposite poles. Am I clear to you? And during the telophase 1, nuclear membrane and nucleolus reappears. Cytokinesis follows. So this cell is this is called as dyad of the cells because there are two particular nucleus in the same cells. Okay, now the stage between two meiotic division is called as interkinesis and it is short lived that follows a prophase 2. The stage between the two meiotic divisions andre meiotic 1 and meiotic 2 between alli one chikka small short lived phase irutte, adana navu enanta karitivi, interkinesis. We call them as interkinesis. Meiosis 2, it is equal to your mitosis. Remember this. Meiosis 2 is equal to your mitosis. Am I clear to you? So it is initiated immediately after cytokinesis before chromosome gets elongated. Okay, so cytokinesis agid takshana meiosis 2. Okay, cytokinesis of what? Telophase 1. Okay, in prophase 2, nuclear membrane, same thing children. Nuclear membrane disappears and chromosome becomes compact. Metaphase, what will happen? Same thing, they align at the equator where kinetochores of the sister chromatids will also reappear. Now anaphase 2, they split up and the centromere move towards opposite poles. Okay, telophase, again the same thing where it will divide and the nuclear membrane will be reappearing back. So finally over here, what is the point? Nimge meiosis 1 and 2 agidre 2 cells end of the time barutte, correct? So meiosis 2, you will have a 4 cells formed at the end of the day. Okay, so that is the importance of this. Now moving ahead, significance of the meiosis. Meiosis form a gametes that are essential for the sexual reproduction. So for gametogenesis you require meiosis to happen. Crossing over introduces new recombinants of the traits. Okay, so usually this crossing over is required. Why your crossing over is required children? Because your paternal and maternal, when they form or when they overlap with each other, they usually what to say, help and form a new variations. Okay, so this is the one, helps in maintenance of the chromosome number of sexually reproducing organism. Okay, so what happens, it helps in the maintenance of a chromosome number of sexually reproducing organism. It provides evidence of the basic relationship of an organisms. Okay, so the relation between andre paternal, for example there is a child and we need to know whether the child is a parent, this is the parent or not. Why? How do we know? Because of the genetic material present in us. Okay, now this is a small difference between mitosis and meiosis. Takes place in somatic cells. Meiosis takes place in your particular reproductive. Single division which produces 2 cells. Meiosis, after your meiosis 1 and meiosis 2, 4 cells will be formed. Haploid and diploid, both kind of cells may undergo mitosis. Only diploid cells will undergo meiosis. Crossing over is absent, crossing over is present. Pairing of chromosome is not there, pairing of the homologous chromosome is there. So this is your basic difference between mitosis and meiosis. Okay. Now let's start up with your particular what to say, the MCQs. Okay. Now children, read the following statement. Okay. So read the following statement and read the following statement about cell division and select the correct answer. Okay. Now, first one. M phase represents the phase when actual cell division occurs, and the first phase represents the phases between two successive M phase. Second one. 24 hours average duration of a human cell division. Cell division proper lasts only for about an hour. M phase constitutes more than 95% of the duration of a cell cycle. Children, you have to choose which is the correct option. Okay. So if at all you look into, M phase represents the phase when actual cell division, correct. First phase represents the between two successive M phase, correct. Next, in 24 hours, yes, it is human cell division will appear. Cell division lasts up to for an hour, correct. M phase constitutes, which is the 95% of the cell cycle. It is your interphase. So this is your wrong option. So which is the correct option? A. Okay. So option is A. Moving ahead. Identify A, B, C and D. Not all easy there. A, B, C and D, which is the option. Okay. So A, B, C, D, what is it? See properly. A ಏನಾಗಿರುತ್ತೆ when the cytokinesis ಆಗಿದ ತಕ್ಷಣ ಒಂದು ಗ್ಯಾಪ್ ಏನಾದರೂ ಬೈ ಚಾನ್ಸ್ ಕೊಟ್ರೆ ಓಕೆ ಆಕ್ಚುಲಿ ಏನಿದೆ G1 S and G2 correct but since I have given 4 now obviously first one will be what G0 then there is no division at all so G0 G1 and S and G2 so option is your A okay moving ahead. Select the incorrect statement regarding the S phase of the interphase. Okay. Now, moving ahead for this particular thing, incorrect statement occurs between G1 and G2. Yadu S. So correct andre G1 anmal S anmal G2. Correct. Centrioles duplicate in cytoplasm. Correct. DNA replicates in nucleus. Correct. Yes or no? As DNA is double, number of chromosome also doubles. No, it is only replicating. It's not doubling up. Illanidre nam chromosomes alla ಜಾಸ್ತಿ ಆಗ್ಬಿಡೋದು. So option in draw D. Match the column with column 2 and select the correct option. Okay. So I'll give you like half a minute of time. Just see children. What happens exactly? Match the following. So chromosome move towards the spindle equator. Yadrali equator line yaduk hogodu metaphase alli. Okay. Next one. Centromere splits and chromatids apart. Yadalagutte makle it will happen in your anaphase. Yes or no? Because they are moving apart. Correct. See pairing between your homologous chromosome. Where is the pairing of the chromosome happening? It is in your zygote. Crossing over between homologous chromosome, it is in your packaging. So option in draw C. Human cells in culture shows a cell cycle of completed approximately how many hours? Children, look into the particular option. You have two options which is correct, that is your B and C. So both the answers are also correct, 24 hours and 24 hours. Okay. So B and C is the option. Next one. Each the cell divides once in approximately every hour 90 minutes. Am I clear to you? So option is what children? 90 minutes. Okay. Next one. Which one is the correct sequence of the cell cycle? Okay, which is the correct sequence of the cell cycle? Makle bega nodi easy ide. First one yadu G0 ide adralla nodಕೊಳ್ಳಿ ಯಾದ್ರಲ್ಲೂ ಇಲ್ಲ so ಅದಾದ್ಮೇಲೆ ಏನು ಬರುತ್ತೆ G1 S G2 and M phase. So which is the option? Option is your C. A cell cycle includes what and all it will include? 8th one. Okay. Interphase, metaphase and divisional phase. Correct. Metaphase itself is called as divisional. Prophase, metaphase, anaphase, telophase. Ashtena. Where is interphase? It will also include interphase. Yes or no? So interphase is not told. Only a mitotic phase is told. So this is the wrong option. Okay. Okay. Next up. G1, S and G2. Where is mitotic? Mitotic is not there. So again this is wrong. Karyokinesis and cytokinesis. Where did the rest of the things go? So the answer is in okay. A. Which of the following phase of cell cycle is not a part of interphase? Yadro easy. G1, G0, S. So yadalla interphase though. It is your mitotic phase. Yes or no? Now, to build up a food resource in the particular cytoplasm, chromosomes become unfolded to start your transcriptions. I'll repeat again. To build up a food reserves in the cytoplasm, chromosome become unfolded to start a transcription of mRNA, rRNA during which of the meiosis one? Okay, so during the which of the meiosis one it will start. Children, what did I say? You diakinesis is the last one. So leptotin is the first one, zygotin where you have your homologous pairing and all those things. So what is the point? Where did that particular synthesis begin? It was in your diplotin stage. Am I clear to you, children? Now moving ahead. Non-dividing cells of the heart cells and the nerve cells will dashly go into ಹೇಳ್ದೆ ನಾನು ಏನು ಮಾಡಲ್ಲ ಏನು ಇಲ್ಲ ಅಂದ್ರೆ ಯಾದಕ್ಕೆ ಹೋಗುತ್ತೆ definitely it is your G0 phase. Okay, quiescent stage. The cells that do not divide further and inactive, again what is the option, children? Easy, same answer conro G0 stage. If they don't divide, quiescent stage yado G0. Why? G1 and G2 and S phase. G1 you have your growth happening, DNA synthesis happening in S phase. G2 again there is a growth, but in your G0 there is no cell division, so it is your G0 stage. Now children, centrioles undergo duplication in dash of dash and begin to move towards opposite poles of the cell during dash end stage of a dash. 13th one. Okay, now coming to your 13th one, centrioles undergo duplication during your what phase? S phase. Okay, of which phase? Of your interphase, and begin to move towards opposite poles. Centrioles move towards opposite poles, not chromosome. Palale itkoli amale anaphase anta bardakbitira. Now they are asking about your centrioles, so it will start moving towards opposite poles in what division, what phase? Prophase of your mitosis. So keep this in your mind. They are asking about the centrioles, not the chromosome. Okay, so option A. Next, 14th one. Dash is characterized by all the chromosome coming to lie at the equator of the chromatid connected by kinetochore and the spindle fiber are from one pole, and it's just the chromatid connected by kinetochore to spindle fiber from the opposite pole. So what is the 14th one option, children? Where the equator lies? Chromosomes. Yeah, equator metaphase. Dash is the best stage to count the number and the study of a morphology of the chromosomes. Okay, so the best stage to study the morphology will always be your metaphase. Keep that in your mind. At which stage of mitosis the two daughter chromatids separate from each other, migrate towards the opposite poles, and are now referred to as a chromosomes to the further nuclei. Okay, so what is it? Which one they are going to opposite poles? ಯಾವ stage ಅಲ್ಲಿ ಹೋಗ್ತಿದೆ ಕಂಡ್ರ? Anaphase ಅಲ್ಲಿ. Okay. During anaphasic movement of the chromosome, of dash of each chromosome is or are towards poles and dash of come trails behind. Okay, so neat ಆಗಿ ನೋಡಿ ಮತ್ತೆ easy ಇದೆ ಕಣ ಮಕ್ಕಳೇ ಇದು. Same answer almost representing. Movement of chromosome, dash of each chromosome, centromere. Okay, move towards the poles and the arms of the chromosome right behind. Okay, so option A. Which of the following statement is not correct regarding the culture cell? It prevents assembly of microtubules. It inhibits the chromosome replication. It is an alkaloid. It is called as a mitotic poison. Okay, which is not correct? 18th one, they are asking you which is not correct. It inhibits the chromosome replication. Okay, so it does not inhibit. Now, what is the true about telophase stage? Okay, what is true about telophase stage? 19th one. A chromosome lose their identity as a discrete elements. Okay, chromosome clusters at the opposite spindle identity as a discrete elements. Okay, chromosome clusters at the opposite spindle poles, nuclear envelope, nucleolus, Golgi complex, and ER reform. And 19th one is all of these. Children, telophase ali chromosome lose their identity as a discrete element, correct? Cluster at the point of spindle poles, correct? Nuclear envelope, nucleolus, Golgi complex, everything is reformed. So option is all of these. Last question. Spindle usually persists in the form of dash during the dash of the method of cytokinesis. Not wrong. 20th question. Okay. Now coming to your 20th question. Spindle usually persists in the form of phragmoplast during the cell method of cytokinesis. So option is your B. Okay, children. So with this, we'll go on to the next chapter.